Hello, everyone, and welcome to Lessons Learned from Executing Complex Medical Device Trials, with a special focus on cardiovascular device, presented by MedPace. I'm Rebecca Friend, the publisher of the Life Sciences Group, and I'll be moderating this webinar. Our speakers today are Deborah Schmalz, Senior Director of Regulatory Affairs at MedPace, and Pirooz Shamzad, Cardiovascular Medical Device Director, also at MedPace. You can read their full bios on the right side of your window. I've got just a few technical notes for you before we begin. If you have any trouble reading a slide, please hold and drag the right corner of the slide window to enlarge. Please also disable your pop-up blockers to participate in the interactive parts of this presentation. If you'd like to download the slide deck, please click the purple globe at the bottom of your screen. The webinar will be recorded, and it will be available for on-demand viewing within 24 hours after the event. We're also going to be following the presentation today with a Q&A session, so please submit your questions during or after the presentations using that Q&A box at the right side of your window. All right, I think that's everything from me. Deborah, please take us away.
Thank you, everyone, for joining us today. First of all, I wanted to give a brief introduction to MedPace and specifically our medical device division of MedPace, and then we'll proceed into the agenda. MedPace Medical Device is a division of MedPace, which is based in Cincinnati. MedPace is a global contract research organization with a definite focus in cardiovascular study management. Since our inception, we have conducted over 330 cardiovascular trials, and as you can see by this representation of the areas that we've covered, we basically have conducted trials in all of the major areas of cardiovascular disease. As a device division, we have conducted 47% of those trials, or 47% of the trials that we've conducted as a device group have been in the cardiovascular space. And again, representing that we have covered all of the major areas within cardiovascular disease. And that is the basis for our discussion today, taking the lessons learned as we've explored these cardiovascular trials and trying to talk about ways to do things a little bit differently, a little bit better, so that in the end you end up with a very efficient and efficient clinical trial process. My name is Deborah Schmalz, and I'm the Global Head of Regulatory Affairs and Clinical Operations for the Device Division. I'm actually based in Minneapolis. I've been with MedPace for 2 years. Prior to that time, I spent 25 years in industry at a variety of different companies in Minnesota as the Head of Regulatory Affairs and Clinical Operations. Pirooz?
Thank you, Deborah. My name is Pirooz Shamzad. Thank you, everyone, for joining and spending their morning or afternoon with us. I've been with MedPace for a year now. I'm Medical Director in Cardiology and Cardiovascular Device. I came directly from the bedside, where I continue on with some academic pursuits as faculty member at University of Pennsylvania. So with that, we'll get on with the agenda and kind of get into our topic for today. So we thought we'd start off with discussing study endpoint selection. As everybody knows, and again, it may seem a little basic, but I think sometimes we forget that the success to the end is really understanding the basics. And so, forgive me if some of this does seem somewhat mundane, but a clinical trial, you have to remember, has two main goals and with respect to regulatory approval, and that's to prove, one, efficacy and to prove safety. And so everything we do to design the trial and to design the endpoints that assess the device need to come back to these two main points. And in so doing so, there's a checklist of endpoints that I would consider whenever when I'm trying to think of what's the best group of endpoints to choose. And a lot of this does come from the FDA guidance, and I've adopted this from that guidance. And so the 5 main things that I look for, I think, is number 1, that the endpoint successfully characterizes the clinical effect. I think that's an important thing, of course. That gets back to the efficacy of the device. Importantly, there needs to be clinical meaningfulness as well as relevance. If there isn't, then really how is that device going to be applied in real world and in practice? There has to be clinical benefit. If without it, there's no reason to use the device, of course. It needs to be an objective measure. One, for not only the study, because it needs to be reproducible, it needs to be accessible, and it needs to be scientific, but then also in practice, it needs to be a way that the clinician at the bedside can objectively measure the success of the device and compare to what the trial ultimately showed. And then it needs to be readily accessible. This is, I think, important and frequently overlooked. While some sites, maybe a large academic center, has a luxury of multiple modalities, say in imaging or in a procedural aspect, not all sites may have that. And if your design of the trial, the endpoint, relies on a very specific modality that not every site can employ, that may render your trial ultimately limited to a very small set of sites. And ultimately, if you were to get approval, it will ultimately render your device limited to a very small subset of sites, which is not, I think, the goal for the majority of folks. So on top of this checklist of endpoints, or successful characteristics of endpoints, comes another layer of unique challenges that are specific to device trials. And this plays into how this checklist of successful characteristics needs to be interpreted. As most of you know, device trials are typically a single pivotal trial following a feasibility trial. And this is differentiated from a pharma trial, which may be multiple feasibility or extended phase 2 process, where then at the final phase 3 has the luxury of multiple assessments, multiple trial and errors, if you will. Most trials in device are of smaller sample size. They're of shorter study duration and shorter follow-up duration. There's typically difficulty blinding given the physical characteristics or extracorporeal nature of the device. There's typically operator dependence. Someone has to either implant a device or operate the device, which then has a learning curve, which then gets back to the reproducibility or objective assessment of the endpoint. There's typically modifications in the investigational product, less so in pharma, more so in device, where you will have generational changes or modifications / improvements, which may ultimately affect not only reproducibility of the data, but comparability of the data. And then another whole bucket of discussion would be combining a device with a drug and all of the challenges that come with that. So adding these several points to the checklist then kind of basically results in a very diverse set of endpoints that one can utilize in a device trial. As I move forward, you'll notice that I won't specify, here are some key endpoints, these are the endpoints you must use, because again, it is diverse. It really depends on the product, it depends on the population, it depends on the goal of the trial or the regulatory pathway of the trial or of the device. And so I'll speak to some generalities and really do this in the form of case studies from recent projects that we've been involved with. So our first case study is an interventional cardiology trial. It's a pivotal trial, and the challenges here were twofold. Number one, there was an inability to assess the primary safety and efficacy endpoint. And there was really for two reasons. One, the assessment was not standard of care, and two, the study duration was too short to assess the treatment effect. And the second challenge that was faced, the study was not powered to adequately assess secondary endpoints. So kind of tying back to some of the points I made in the previous slide. So when we talk about sample size, clearly we all understand that sample size should be determined to power the primary endpoint. If there are secondary endpoints or exploratory endpoints that are needed, that needs to be accounted for in that sample size. So clearly it means a larger sample size. But that really gets back to the question, what is actually needed? And that's really, I think, an important lesson learned from this experience that we had. And so when we met with the sponsor and I reviewed through the endpoints, really realized that some of these secondary endpoints were not really needed. They were nice, they were scientific, they were probably better served as a sub-study. And so really kind of thinking what needs to be in this current trial, what gets me to efficacy and what gets me to safety versus what's just nice to have. Those nice to haves, think about a secondary study. And then the third point that I think gets overlooked is that not all sites are the same. So practice variation will dictate availability of the assessment, which limits the usage, as I mentioned before, as an endpoint. And it also limits the ability to utilize the device post-market, post-approval. Standard of care assessments. It's usually best if you can find an endpoint that aligns itself with the standard of care assessment, because the sites are going to be, one, used to doing it, apt to do it, they have the resources to do it, and the data will probably be consistent because they are well versed in it. That's not always the case, especially if it's a novel device with a novel endpoint, then that may not be the case. And so if you aren't able to align yourself with an SOC assessment, make sure that protocol assessments are performed rather than reversion back to SOC. What that means is continual site communication, site training, really understanding what the protocol assessment is, how to do it, and making sure that we don't get lax in querying the quality of the assessment. And lastly, study duration. As I mentioned before, device trials typically are shorter, and so we really, when choosing an endpoint, we must be careful to choose endpoints that can be assessed within that duration. So if you're looking at, say, functional change, cardiac functional change, you may not be able to assess that in, say, 3 months or 6 months. You may need to look at 12, 24, even 36 months out. If you're unable to design that into the study for whatever reason, make sure the endpoints reflect that. If you do have the luxury of extending duration and looking out further, then that's great. But then with that comes the need for extended follow-up, extended site communication to make sure that follow-up compliance stays of high quality. And then again, if you're not aligned with SOC, this becomes amplified in that follow-up period because sites are more likely to go back to what they know as you get farther and farther from treatment day 0. Our second case study is a little different. This was a vascular access device. It was a feasibility study of a novel device. And so the challenge here, and we were brought into this very early into development, so really worked with a sponsor to develop this plan. The challenge that was quickly noted as soon as we saw the device was we'll need a physiologic endpoint to gauge efficacy. However, given the mechanism of action for this device, there was no precedent for such an endpoint. And so kind of the thought process behind that is, okay, number one, the type of study, which was great. This is a feasibility study, so it's actually perfect for us to assess different clinical and imaging endpoints prior to the pivotal trial. This is our chance to really explore what really does capture the efficacy of this device, what actually describes this device best before we're actually in the pivotal setting. I'll take this moment to mention about surrogate endpoints, and I'll do that in the auspice of imaging. So first and foremost, surrogate endpoints are a great thing because, especially in a setting of a unique device or a novel device, you need to be able to fall back on something that we do know to describe what we think is what is currently an unknown. So it's perfect for this scenario. However, in order to do so, there has to be a rationale to providing that surrogate endpoint. There needs to be prior clinical work that at least relates the therapeutic area or the therapeutic effect of the device to what you think is applicable in that surrogate endpoint. When you do choose your endpoint or screen through the surrogate endpoints, make sure that you're utilizing the most standard, most widely accepted acquisition technique. If a single center in the world is doing one assessment versus the rest of the world, that is probably not the right assessment to choose because there's only one guy or one lady or one center doing it, and that's not going to be, that's not going to provide the rationale that you need. By utilizing the most standard, most accepted approach, then you'll have the most buy-in, again, better data, more consistent data to actually maximize site participation and then foster standardization and ultimately give you data that you can use at the end, especially in terms of designing your pivotal trial. So now, speaking of surrogate endpoints, clearly I should probably mention something about composite endpoints. They're quite difficult, not really well favored in terms of their practical use, just given the fact that they are hard to design and are riddled with limitations, but review them nonetheless. So composite endpoints, I feel, are probably best when you are looking at a well-characterized disease, that there's not that many unknowns about the natural history or the symptomology, presentation, etc. With the well-characterized disease state comes generally an agreement of what clinically important endpoints there are at the bedside, which then translate to the efficacy endpoints and safety endpoints for the study. And generally speaking, hopefully there is agreement that all those endpoints are clinically important. The second thing that's important, it's useful, is when the endpoints have an expectation of all moving in the same direction. So say in heart failure, that with increased hospitalizations comes increased mortality. That's important. For some reason, if increased hospitalizations led to decreased mortality, now you have endpoints that stray away from each other, which then renders your composite endpoint neutral. Not helpful. So advantages for utilizing a composite endpoint is that it amplifies the treatment effect, which increases the number of events. That's really why you would use these. Effectively decreases your sample size. And that's really the benefit of a composite endpoint. But the limitations, like I said before, are somewhat numerous and quite important to note. So first and foremost, I think there's a vague interpretability, as not all endpoints may be of equal importance. While all may agree that these X number of endpoints are all clinically important, there's rarely consensus on how important one endpoint is compared to the other. And so really there's no good way of weighting the endpoints. The worst case scenario that could probably happen when choosing a composite endpoint is that the least clinically important endpoint may drive the outcome difference. And that's not going to be helpful in terms of efficacy analysis at the end. And then, again, like I mentioned before, there's a possibility endpoints may trend in different directions, which then renders the endpoint relatively neutral. And I was going to wrap up this section just talking about blinding very briefly, and I'll touch on this again when I talk in the later section about imaging and imaging endpoints. A double-blind study, although ideal, may be very difficult, if not impossible, to accomplish due to the physical characteristics or function of the device in question. So the inability to blind will then, of course, incorporate bias. So mitigating that bias is key. There's an understanding that there will be bias. There's an understanding that blinding is not possible, that the FDA will clearly accept. You just have to think of ways to mitigate it and minimize it. So utilizing objective endpoints is important for the reasons I outlined before: accessibility, reproducibility, and high-quality data. If you can ever use a third-party evaluator who is blinded, like a CEC or a core lab, a central reading lab, that's very important, and that would be very helpful. And then the longer you can keep the study staff blinded through enrollment, screening, and literally up to the procedure, at least there's preferential dropout, or there's minimizes the potential for preferential dropout, and it minimizes any bias leading up to randomization and to determination of the treatment arms. So with that said, I'll turn it over to Deborah, who will talk over some data considerations, and I'll join you again to talk about some imaging considerations.
Thanks, Bruce. What I'd like to talk about now are some of the planning things that you need to take into account as you're embarking on a study. Obviously, we all know that we're conducting clinical trials because we want to have high-quality data that comes out of it so that we can either seek regulatory approval or obtain manuscripts documenting the safety and effectiveness of our therapy. So when you're thinking about the data that you want to capture, we find it's helpful to really answer these 5 basic questions. Who is going to collect the data? Where is the data going to be collected? What data are you going to collect? How are you going to collect it? And why are you collecting it? First, moving to the question of who. This is one of the first things that you really need to understand, especially when you're dealing with a complex trial. You need to factor in whether or not the situation that's being monitored is happening acutely, or is it something that is a routine monitoring of the patient that follows, as Prue said, the standard of care? Who's going to collect that data? Is it going to be, are you going to count on the emergency room physicians or staff to collect the data, or are there dedicated research staff within that environment? These are key questions that need to be answered so that you can design checklists and data capturing mechanisms that fit this particular need. Do you need to have provisions for collecting data on a 24/7, 365-day schedule? Clearly that impacts how you design your case report forms and, again, the collection tools. And then lastly, another question is they're a specific location relative to the subject enrollment that might be different from where the care is actually provided. You really need to understand in this context what is the continuum of care for that patient, where are they going to be flowing from, where are your referrals going to be coming from, and where do they move within the care setting. And obviously, once you've taken that into account, really providing customized training that covers all of the individuals that will be involved in collecting this data. Obviously, that starts at the beginning with a very good and well thought out site investigation or study investigation visit, the investigator meetings, understanding the key players within each of these investigational sites so that you provide customized training. We found it very difficult or very important to provide tip cards, which might help key individuals that are involved in this to make sure that they know when there is a particular assessment that needs to be done. We need to make sure that those tip cards and inclusion-exclusion posters, all of those nice training tools and study tools are in the right location and touch the right people that will be involved in the trial. And also, if you are dealing with a trial that does involve the emergency room, such is often the case when you're dealing with cardiovascular trials, make sure that you in-service all of the emergency room department staff members that might have the ability to identify patients that are potential candidates for your trial. Next, moving on to where is the data captured. Again, really thinking about what that patient flow is. Where will each of these events be taking place? Do you have within each individual visit data that's coming from a variety of different locations within the hospital? So you need to understand, is this something, again, that's happening emergency, or is it on an emergency basis, or is it just urgent? Are there ways for screening to have auto notifications when a patient that is a potential subject is brought in and admitted into the hospital? From a post-procedure assessment perspective, you really do need to understand if there are regional differences that take place not only within the United States, but also geographic differences. So if those post-procedure assessments take place remotely from where the patient originally entered the study, you need to make sure that all of that continuum is taken care of. And more specifically, even within the hospital environment, understanding the differences between data capture that takes place in the CCU, the telemetry unit, step-down unit, the cath lab, all of those things need to be taken into account. And then lastly, moving into the long-term care follow-up, again, understanding whether or not you need to engage and train not only the cardiologist that's doing the primary interventional procedure, and then also the primary care physician, which may be involved in the longer-term follow-ups. Oftentimes we found, too, that happens at a remote location, so you need to make sure that you consider that with respect to all of your contracting, with respect to IRB considerations, too, so that you ensure that you have all of these individuals and all of these entities involved in the data capture portion of the clinical trial. Next, moving on to answering the question of what. And Peru's talked about this earlier. It's really important when you think about the what and the data that you're capturing, the differences between standard of care and the per protocol assessments. This drives many things, obviously, but the institutions do need to be aware of that because there may be extra costs associated with those non-standard of care assessments. These may differ per hospital. It may become more difficult to actually capture those non-standard of care assessments, so just make sure that there is a complete understanding of that protocol. Again, starting with the SIV visit through the protocol training, each and every member of the staff at that investigational center does need to understand what's the norm in terms of patient care and where those differences exist. if at all possible, in this clinical trial, when you're selecting those endpoints, make sure that you're avoiding duplicate tests. Not only does that sometimes mess up the patient flow within those investigational centers, but it also does add costs, and sometimes, obviously, when things are very, very busy and you're dealing with an acute situation, if you're asking an individual to perform both an angio and an IVUS, when from a clinical perspective only one of those is required, physicians and investigators dealing with an emergency type situation may defer to what's most comfortable for them, and we don't want to miss that key point. So again, avoid it if at all possible, but if you do need to have duplicate tests, make sure that the institutions are well prepared and understand the implications of asking for both of them. From a measurement consideration, and this is something that is of interest not only geographically within the U.S., but also from a worldwide basis. If you are dealing with a global trial, you need to make sure that all of those standards that are being used for measurement are universal, that the units are understood, that if there's a lab test that's being done, that everybody has an understanding of how that test should be done, what the units should be reported, what the normals are, and on from there. Again, the goal is to take into account all of these differences so that you have a standard set of data that can be analyzed at the end of the trial. Next, moving on to the how. So now that you've got all of this figured out, and probably in concert with those previous questions, really understanding how best to capture that data. Obviously, we have two choices at this point in time, paper versus an electronic data capture system. Most people at this point in time are probably using electronic data capture, but yet there are certain situations where paper might be a better choice, especially if you're dealing with situations where internet access might be spotty. You could consider paper. Cost is a consideration, although most of the differentiators now between paper and EDC have gone away, but think about ease of use too and the complexity of the data that you're capturing. Regardless of whether or not you go with paper or EDC, especially for complex trials, give consideration to data collection aids, especially when you have patients moving through multiple places within the hospital with non-standard of care assessments. Consider some color-coded aids that guide the research coordinator as to what's required at what point in time. And then for things that are non-standard of care or even for the routine follow-up, consider creating standing orders for those situations so that people aren't rushing at the end and you miss a critical time window because an order was not available for an assessment. And last, think about where your source documents are and whether or not you need to provide worksheets, diaries, or device logs to capture important data. And last, why? Why is it important? Obviously, the goal of all of the work that you do up front to plan data capture really is intended to drive at assessing your primary endpoint from a safety and efficacy perspective. Consistency in that regard is absolutely crucial. And if you don't have consistency either with the way that the data was captured or that you're missing key data points, you will render the study less useful. You may miss your endpoint because of lack of consistency or failure to capture data appropriately. And obviously then, secondly, from a secondary endpoint, as Prue has mentioned too, make sure that you're considering that, but don't be so focused on those secondary endpoints. Consider them as sub-studies if they're not really important for determining the primary endpoint. Especially in a complex trial, you don't want to mess things up because you're asking for questions to be answered that maybe are better done in a post-marketing type situation. Last, a little bit about monitoring. and making sure that you take into account certain things as you're developing your monitoring plan. From a combination products perspective, make sure that you understand the regulatory implications, and especially as it relates to safety reporting, and especially if you're doing a global trial where you have to factor in the various time points that are required for various safety reporting requirements to regulatory agencies in Europe or abroad. Oftentimes they are divergent, especially if it is a post-marketing type situation in other geographies. Make sure that your safety team understands what they need to report and when they need to report, and so that you don't miss a key deadline for those reporting requirements. From a setting perspective, it's important to understand, again, where is your data going to be monitored? Is it hospital versus clinic? Do you need to make sure that you take that into account as you're developing your monitoring plan? And then also from a regulatory coverage perspective, understanding again that from a monitoring perspective, if it's pre-market in the United States and post-market in the EU, making sure that you've got plans in place to ensure that the EU component of your study is just as sound and just as concrete as your U.S. component. And then lastly, just a little bit about risk-based monitoring. Obviously, this is relatively new to the regulated industry in the United States. I think the jury is still out about how this is going to shake out, but certainly give that a consideration. Key elements there are the development of a proactive risk mitigation plan, determining endpoints, or not endpoints, excuse me, key criteria that will trigger certain events. Key to that is to ensure that if you develop a risk-based monitoring program, that you are routinely and on a real-time basis looking at the data that comes in, so that you don't wait for 3 months to realize that you missed a key point that should have driven a risk-based monitoring visit. Bottom line, it has to be a dynamic process if you are going to use risk-based monitoring. With that, I'll turn it back to Farouz to talk about Imaging Core Labs.
Thanks, Deborah. So as promised, I'll talk a little bit about Imaging Core Labs, and really in the auspice of imaging endpoints, and how some of the considerations in the first section I presented can be ameliorated by using an ICL. So when we talk about imaging endpoints in cardiovascular trials, it's important because they're commonly used. More and more, there are targeted modalities that assess procedural outcomes, and then there's multiple modalities that have been around for quite some time, and new ones that come around every so often that assess functional changes, like heart function. The issue, though, becomes that there are a number of factors that can then impact the ultimate utility of that endpoint, and that is trial-specific imaging protocols and charters. How well is that charter designed or written, and how well can sites follow it, and how good is the data that comes out of that protocol? Non-standard assessments. We kind of touched on this both in Deborah's section and in my previous section. If you're forced to use something non-standard, how are you going to improve reproducibility? How are you going to make sure quality and standard are existent in the data? Uniform assessment speaks to the same issue. Blinded interpretation. You've got a trial that perhaps you cannot blind, as we discussed before. This is a good opportunity to invoke some sort of blinded interpretation to help reduce bias. And then quantitative measurements. If you have to measure something, then you introduce more bias, you introduce the more type of error. This is hopefully a way that we can help assess that and address it by utilizing an ICL. So before I go further, though, I just want to kind of set the stage of what I'm talking about exactly. So some trials may require a single qualitative assessment of a basic, well-accepted modality. I am not talking about those trials. Those trials probably do not need the added expense and added structure of an imaging core lab. A good example, and this comes directly from the FDA guidance, is that of a bone X-ray. So if you're doing some type of orthopedic study or calcium manganese study, and you are looking at the number of bone fractures that occur during the study period, a simple X-ray of an arm probably does not require an ICL. But when we start talking about complex cardiovascular trials, we don't have that luxury of a single bone X-ray. There's advanced imaging modalities. Now you're talking about nuclear imaging. You're talking about advanced cardiac MRI. You're utilizing almost always quantitative measurements. It's not just is bone broken, yes or no. Is there a cardiac dysfunction, yes or no? And if yes, then what is the degree of that? Quantify that for me. And then most of our trials are going to be global, or at least there's differing practice patterns among a region that is selected, which then all introduces changes and differences, variation in the data, which ultimately subtract against the end analyses. So going back to that case study model, this is a different interventional cardiology trial. This is a global pivotal study. The primary imaging endpoint, which was a functional assessment, was to be quantified at the site level. And so early in the startup process, the sponsor became it became evident to the sponsor that not all the sites were adequately equipped to properly quantify the measure. Not all the sites have the equipment, astonishingly enough, and that, although was asked in early feasibility, was missed. And then the sites that did have equipment, not all of them had the ability to process the imaging. They didn't have the right back-end software, or they lacked the expertise to accurately or reproducibly measure. This was not asked for in feasibility. It was overlooked. And so you had 3 or 4 readers who did it differently or hadn't done it enough to develop a systematic way of doing it. And so even in that one center, there was such inter-observer variability that data was rendered useless. So for the sites that did provide data for some of the reasons above and others, there were a wide range of values that came in the initial data set that just made absolutely no sense. And quickly the sponsor realized there was an issue, at which point we were involved, which is at least good for the sponsor. So this is exactly where I think an ICL can be very helpful and touches on some of the facts or points that I highlighted early in this slide deck. The ICL, I think, bottom line, affords a study-wide efficiency when applied to the appropriate situation. And so really in multi-center global trials where you can expect wide center variability, imaging acquisition and the endpoint is not well standardized, again, for the fact that there probably will become center variability or data variability. If quantitation of the image is required, that's just going to help reduce the amount of error among the data points. And then if the study is unblinded, as I mentioned multiple times now, it does offer a mechanism of blinding, which will be looked well upon come time for approval. Beyond just some of the mechanical tasks of reading, the ICL can provide a couple of number tasks, which then will ultimately help data quality. I think first and foremost is efficient startup. Feasibilities. I think the ICL will know what questions to ask. They know, and not just in terms of equipment, but in the software, in terms of the reading, what's required of the readers in terms of training or prior experience. All things are important. Developing a charter imaging protocol is quite important because that's going to dictate how the sites move forward and how they will conduct the assessments. And then once all of the beginning stuff is created, then really developing site training, developing qualification, and developing equipment management plans to make sure that the study continues on forward in the right path. Once the study is kicked off and startup is done, then continual management of the above is required to make sure continual performance. Consistent data collection can be verified and uniform interpretation can be verified, and that's via regular sampling of any data trends that comes through, offering blinded readers for reasons mentioned before. And then for some ICLs, they actually can provide a service where they can qualify subjects for inclusion and exclusion. They can actually get built into the randomization scheme and assist in assessment of inclusion-exclusion criteria. So with that, I'll kind of turn over back to Deborah, who will kind of finish off the morning with some regulatory considerations.
So by now you've probably all read and memorized the new draft guidance document that FDA recently issued about the use of foreign data to support U.S. approval. And I'm not going to go through that in depth, but I look at this and I say, really, what is this draft guidance talking about? It's talking about how, as device sponsors, we maintain the device data integrity and relevance to the U.S. approval needs. There's some very good information in this, and if you are in the situation where you do want to use foreign data to support your U.S. approval, I highly recommend you go through this. And it is still a draft guidance document, but yet there are some very relevant points in there. Key things too, if you're thinking about that and the use of non-U.S. data, make sure that you are talking to FDA about your intention. Usually this is not going to be a big deal, but I would be remiss in saying that you just always assume that FDA will accept that foreign data. So then thinking more specifically about a very specific situation where you have a device that already has a CE mark and you are now trying to secure U.S. approval and you want to make use of the investigators that you have in Europe with experience in the device. Some of the things you need to think about in this situation is making sure that whoever you are using to secure the approvals in the European countries, whether it's the ethics committee or the competent authority approval for conduct of that clinical trial, fully understand the U.S. law and don't make requests for changes that are inconsistent with the regulatory requirements. We have seen certain situations where, for instance, the German competent authority or ethics committee of BfArM will ask for changes to the informed consent that are completely inconsistent with what the U.S. requirements are. So make sure that you understand that, and it's not to say that the consents have to be consistent across the board, but you need to make sure that they're not asking for changes, perhaps even in the inclusion-exclusion criteria that will jeopardize the ability for you to use that data to support your U.S. approval. Secondly, make sure that you take into account study startup timelines, and this is just a heads up for any of you that are considering a complex cardiovascular trial using Germany, for instance. We know that the study startup timelines in these situations, even for post-marketing situations, are dramatically extended at this point in time, especially if you are using non-standard of care radiation assessment or radiographic imaging that's not standard of care, because that has to be reviewed by the German Radiation Safety Committee. At the current time, there is a tremendous backlog with this particular group, and we have seen some studies delayed from startup in Germany by 6 to 9 months, just because of the backlog with this particular review committee. Last, and we've touched on this a number of different times, for those European investigative sites, they have to clearly understand that this is not just a post-marketing trial. If you do not set this up correctly at the beginning, you are going to end up with not a disaster, but something that's very difficult to manage. We have seen situations that sponsors have brought to us where their European investigational sites did not understand this. They missed gathering important data. The visit schedule was just totally ignored because they were really viewing this as a post-marketing study. So it's important for those centers to understand that the tenets that are called out in the protocol are not optional, and that this is a pre-market study for all intents and purposes. So now moving on a little bit to enrollment and consenting, and this actually extends throughout the course of the clinical trial program. So some things to think about there. We all know that the worst thing that can happen to you as a clinical trial manager or VP of clinical or director of clinical is that you plan for a study to take place in 18 months, and all of a sudden you're in the situation where your enrollment is lagging. You've got investors potentially or board of directors saying, what's up with this? How come the study is dragging on? So how do you deal with this? We've tried today to talk about all of the different things that can happen. You might have inconsistent screening. You might not have the ability to train the sites, or you didn't train the sites correctly. You've got enrollment that's missed. You've got issues with the treatment protocols being not aligned with standard of care. The impact of that is you end up potentially with a very chaotic situation. How do you avoid that? What we really are advocating is that as you're setting up your clinical trial program, before you enroll that first or engage that first site, really do a good job of identifying risks associated with this protocol and manage them consistently throughout the process. If you do a good job of that, you're going to end up with a much better enrollment that's predictable, and obviously end up with a more efficient execution of your clinical trial. So just some examples here from a case study. In this particular situation, this was an interventional cardiology trial. It was a pivotal trial that was designed to secure U.S. approval. The patient population was interesting because there was a very, very short window between symptom onset and treatment that was allowed. And that worked against us in the sense that the investigative process would interfere potentially with the door to balloon time. The second factor here was that this was obviously an emergent patient population which could present any time, any day of the week. So the lessons that we learned in this particular case is how do you capture those patients? Obviously, as we said earlier, you need to understand what the research staffing constraints are. You need to be able to capture and make use of some of those emergency room staff to identify potential subjects, and simplify and stage the screening if possible so that you minimize that door to balloon time. So what I mean by that is make sure that you understand where those data points actually must be captured, and if there's a way that you can delay some of those until after the patient's immediate needs have been taken care of, that's going to make your trial much more easy to manage. Secondly, with respect to consenting, make sure that you consider in this type of situation the use of the two available options that are provided by FDA, and the first is the short form informed consent, but which really allows you to read to the patient a shortened consent under as long as the witnesses are present, and then have them sign a short document. And then potentially also consider the emergency use exemption. So just to sum up, again, with respect to this section, one of the things that you might want to consider is borrowing from our friends in quality a very formal risk assessment plan and risk management plan that identifies risk categories, what the description of the risk is, and then also very conscientiously saying what's the probability of that occurring, the impact to the study, so that you end up with a risk management score. Obviously, you want to pay attention to the areas where your risk management score is the highest, because those are the ones that are most likely to occur and have the biggest impact to your program. When you're doing this, make sure you have well thought out what your mitigation plans are, your contingencies, and who's actually responsible for looking at that and determining whether or not you're on track or off track. These types of tools can be very useful throughout the course of the trial, during your planning, and can assess very critically each element of the clinical trial program to make sure that you've got a well thought out execution plan in place. That is the end of our formal presentation, and I understand now we'll be open to Q&A.
Okay, great. Thanks. Great presentations, everyone. So time to move on to Q&A. We've got a lot of questions. We'll try to get to as many as possible in the time that we have. So here's the first question for you, and this can be either one of you. Feel free to jump in on this one. Why do many device companies start with EU approval?
I'll take that one. I think there are a couple of factors that are in play there. First, generally speaking, although this is changing slightly, the barriers to approval through the CE marking process are usually much less than the FDA. especially now that FDA, for many devices, is trying to pigeonhole them into the de novo route, which is potentially a good thing, but the regulatory pathway is a little bit more complex. Secondly, is the fact that many of the notified bodies in Europe generally require less clinical data than the U.S. FDA to grant approval. So those are the two main reasons.
Okay, great. This is another question for you. How do sponsor field representatives impact clinical trials in the device landscape?
Oh, that's a great question. Your sponsor field rep can either be your best friend or your worst enemy, and so some things to consider as you're dealing with a sponsor field rep is to make sure that it's well understood what that rep's role is. And that's going to vary based on what the device is and what the clinical trial is. So unless you have roles and responsibilities well defined, that sponsor field rep is going to be the bane of your existence as you're managing that trial. Where they can have a huge impact is obviously with devices that are very technical in nature and require training to administer appropriately. That's where they are the most helpful. Secondly, they can help keep and maintain investigator interest in the trial.
Yeah, and just to kind of piggyback on what Deborah said, I think the field reps are, and especially as an investigator, are irreplaceable when it comes to those more novel devices or those that are highly technically dependent. They bring with them the prior training that's really necessary to assist the investigator in properly placing the device or operating the device such that the data that comes from that case is actually usable. So more cases than not, I have found them to be quite useful.
Okay, great. Another question for you. How do you incorporate health economics into your clinical strategy design?
I'll start with that one, Bruce. That's a very good question also. Thanks to the audience for bringing that up to us. That can be a little bit tricky, and it really depends how you are going to use your health economics. On one hand, we all know that in the United States, reimbursement planning is essential, and so you have to factor that in. But on the other hand, you don't want to make your studies so complex that you end up with a longer enrollment time because you're asking for things that are nice to have. So you can incorporate it, but be very, very careful and make sure that you're not increasing the complexity so that you end up with a long trial that you don't win.
Yeah, I think and given the kind of payer reimbursement controversies that have plagued us and then will continue to plague us, I think it's become more important. I think it will become more vital to adding them to the studies. So kind of we'll have to figure out ways to introduce them into studies, but preserving really the primary endpoint and making sure that we don't bog down the primary goal of the study, which is approval. So.
Okay, great. Next question for you. What if the feasibility study does not provide the data to power a secondary endpoint? Can patient numbers be altered after initiation?
Whoa. Okay, so if I understand the question correctly, you've done a feasibility study, but maybe the results of that were not sufficient, so there was either too much variability within the data, in which case that's going to be difficult to manage.
So I guess part of the question I'm unclear is initiation of either the feasibility or the pivotal. So let's answer both, I guess. So if you're starting the feasibility study and you are finding that you're not developing data that can sufficiently answer I'm assuming the exploratory secondary objective, you're trying to assess an endpoint for future work, one has, I guess, two options. You could amend the protocol and explain as to why you need to increase the sample size to power that secondary endpoint. That would probably be, I think, probably a little dicier approach. Probably the more sound approach is to conduct a separate study, a study dedicated to looking at that endpoint and power that study geared towards, I'm assuming, a secondary endpoint that you had your eye on, or maybe there's a beginning of a signal, the data just doesn't get you quite there. Unfortunately, yes, it is a little cost and probably timeline, but probably the more sound way is to develop a secondary feasibility study. Now, when you get into your pivotal trial, if you haven't done the proper legwork, I think you're probably a little bit more stuck or bound there, because now you are in the pivotal situation and that data will be scrutinized. So there probably it would be very difficult to alter the numbers after initiation. You will pay a statistical price because you have essentially reviewed some sort of data to assess the secondary endpoint. That would probably require some consultation with the agency and a determination of what that penalty is in terms of sample size. You probably have to be a larger sample size than what you think you need for the secondary endpoint to assess or to account for the statistical penalty of highlighting this issue in the first place. Hopefully that answered the question.
Perfect, great. And I think we've got room for one more question. So this will probably be our last question for today. And just to let everyone know, if you submitted a question and we didn't get to it, we'll be getting back to everyone after the webinar. So here's the last question for you. What is the biggest challenge for medical device clinical trials across the world, and how can this be overcome?
I think the one that jumps to me, and the reason why when I asked, what I wanted to talk about today, I think it is the endpoint. I think for me, really understanding what endpoint is the proper endpoint, especially in a field that is continuing to advance, modalities, imaging modalities, surgical modalities, percutaneous modalities, all of these are advancing. There's new technology available every day, and so it raises two issues. One is what is currently the best endpoint or best modality that I can bring to the table to assess the product in question. That's hard enough. And I emphasize the word current because now in 6 months or 1 year when my study is done enrolling and I'm looking at the data and now there's new data suggests that one of two things: my current endpoint is actually useless because new data has shown that wasn't very good, or that there's a newer endpoint that's even better. What do I do with that data when I walk up to the FDA with an old, quote-unquote, old endpoint? I think that's really what the challenge is, and overcoming it is going to be hard. I think it's going to be just continual assessment, continual discussion with regards to science and advancing science. I think one of the positive things, though, is that in dealings with the FDA that I've had, as well as the literature that has come out of the FDA, there is an understanding that science improves, that science advances, that there is no one right answer, or what we think is the right answer may change. And so I think open dialogue and continuing and continual assessment of where the study is with the agency will kind of help overcome some of those challenges, at least from a scientific standpoint.
Yeah, absolutely. And just to add one more thing to that, I think, too, it comes back to the age-old question about what's the nice to have versus what is the absolute, not minimum, but what are the actual regulatory requirements to get a device approved and on the market. And sometimes those are divergent for the reasons that Peru's just mentioned. And so you do need to be paying attention to that, but not get swayed into necessarily just saying, okay, I'm going to try the latest and greatest thing and incorporate that into my trial because it sounds nice. So just make conscious decisions about the endpoints that you choose.
Okay, great. Well, again, that's our last question for us today. Thank you both. Really great presentations, and thank you for answering so many good questions. I want to thank our audience for attending this Fierce Live webinar, and I also want to thank our speakers for participating and MedPace for presenting today's webinar. The webinar has been recorded. You'll be able to access the recording within 24 hours on the same page that you used to register for the event. Thanks for joining us, and we look forward to seeing you at future events.